All right, guys. Today we're going to talk about DNA replication, and this is a wonderful picture that is filled with so much information, and I hope it makes more sense to you at the end. All right. First, we want to recall nucleic acids. Remember those that we studied when we did macromolecules? The specific nucleic acids are the macromolecules that carry our genetic material or DNA, right? DNA contains genes, and the genes are the blueprint or the instructions for making proteins located at certain point in the chromosome. Proteins carry out all cellular activity, and if you guys recall, there are two types of nucleic acid: deoxyribonucleic acid, DNA, and ribonucleic acid, RNA. And as you can see in this picture, DNA is a double-stranded molecule, RNA is a single-stranded molecule. So the nucleotide is the monomer for the nucleic acid. It should really feel like a throwback again to macromolecules. The nucleotide has three parts: a sugar. In the case of DNA, that sugar is deoxyribose. In the case of RNA, that sugar is ribose, right? A phosphate here in purple, and nitrogen bases. And in DNA, those nitrogen bases could be A, G, C, or T. But in RNA, those nitrogen bases are A, G, C, not T, but U. Okay, so there are two differences, well, more than two, but based on this, there are two differences between DNA and RNA. DNA has a deoxyribose sugar, and DNA has a thymine, while RNA has a ribose sugar and a uracil. There are DNA base pairing rules. So as I just said, there are four kinds of nitrogen bases found in DNA: adenine, guanine, cytosine, thymine. And to really make it short, what you need to understand is the shape of adenine is complementary to the shape of thymine, and the shape of cytosine is complementary to the shape of guanine. And so these things pair together. ATGC. I'm gonna quote Jacari, if you guys know him. One of the mnemonics that he used to remember this was at Golden Corral. So ATGC, at Golden Corral. If y'all could come up with a better one, come up with it and use it to help you remember the DNA base pairing rules. The structure of DNA, as I said before, it's a twisted ladder, a double helix, right? It has a sugar and phosphate backbone. So this part here that we're pointing to, this light blue part, that's the backbone. So a lot of times you get asked questions, they'll show you like a blank picture of the DNA, and they'll ask you to identify the backbone, right? The nitrogen bases bond in the middle with weak hydrogen bonds, and it is very important that these bonds are weak. And I'm not gonna say why just yet, but I want you guys to be thinking about that. Why is it necessary that the bonds that hold DNA together in the middle are weak? Every other bond is a strong covalent bond, and if you guys remember earlier in the quarter, we talked about different types of bonds. RNA has its own base pairing rules, right? It's a single strand of nucleotides with exposed bases. RNA bases bind with DNA bases with a different set of rules, by a different set of rules. A's binds with U's because we have that other thing that I'm not going to say, and G's continue to bind with C's. All right, so again, think of a mnemonic that can help you remember this, because this is going to be very important for you to remember. Fill out this table. I took out the RNA because I want you to do that on your own. I did the DNA because I want you to see what I'm looking for. So what types of nitrogen bases are found in RNA? What type of sugar is found in RNA? And what is the shape of RNA? All right, so how is RNA different from DNA? So fill out that other side. I'll be checking for that. So DNA replication. So when a cell is ready to divide, it has to copy its DNA. We just talked about the cell cycle. So we're kind of breaking down what exactly is happening in that interface of the cell cycle. So as we remember, the cell cycle is the process of make an identical copy of DNA. That's called DNA replication, right? This occurs in the nucleus during the S phase, the synthesis phase of the cell cycle. It occurs before PMAT, so before actual mitosis or meiosis, whatever is going to happen next. And this is necessary so that each cell can have its own full copy of DNA, right? DNA replication ensures that each new cell has exactly the same DNA. So DNA replication is basically going from DNA to DNA, using DNA to make more DNA. All right. So a parent DNA makes two exact copies. I should tell you that it's semicon. Okay, there. All right. So in DNA replication, the first thing that happens is that the DNA is unzipped. All right. Again, be thinking about why is it that we had weak hydrogen bonds in that middle, right? Enzymes. So enzymes help find complementary bases and bind them according to the DNA base pairing rules. Let's see if I can get this over. According to the DNA base pairing rules, A to T, at, and G to C, golden corral. All right. Two identical DNA molecules are formed with each an old strand and a new strand, right? So when the DNA unzips, each original strand acts as a template. And again, you use the DNA base pairing rules to add on the new strand. So each new DNA has one new strand and one old strand. And because of this, DNA replication is said to be semi-conservative, all right? Is that it? All right. That looks like that's it for us today. All right. That was a quick one.